So did you mark page number 105.6, Modern Synthetic Theory of Evolution, and 5.13, Human Evolution. Only these units are going to be asked for your exam. So we will have a short revision. Yesterday we had seen gene pool. Gene pool is defined as sum total of all the genes present in a population. So these are all the genes. For example, allele AA, then small letter, small alphabet, small a, small a. So all these are the alleles. And all the genes present in population, this is the population. At one time, it is called as gene pool. Not all the individuals will be breeding at a given time. Population may have a distinct geographical boundary. Each individual is a carrier of a part of total genetic complement of the population. So yesterday we had seen about Mendelian population. So the group of individuals which interbreed among themselves, it is a population. So Mendelian population, it is a small interbreeding group of population. It is a small interbreeding group of population called as Mendelian population. So in this we had studied about gene pool. Total genetic information present in the Mendelian population is called as gene pool. So a particular geographical boundary is there. Yes, sir. And each individual carries a particular total genetic complement of the population. See here, gene frequency. The frequency of any given allele. We had previously studied what is allele. I had told you many times about allele. So pair of allele is present. For example, while studying the different characters of pea plant, we had studied about a pair of allele which tells about the character. For example, height of the plant, tall, capital T, capital T. These are alleles of each other. Okay. Then capital T, small t, these are also alleles. But these alleles, this allele, it shows hybrid tall plant. Then this capital T, it shows homozygous or pure tall plant. And small t, these are also alleles of each other. And it is for small dwarf plant. So we had already studied about alleles. So the frequency of any allele, given allele in a population relative to all other alleles at the same locus. Same locus means at the same position. As same locus, locus means position, is known as allele frequency. Allele frequency is equal to number of copies of a particular allele in a population. You have to remember this, number of copies of a particular allele in a population upon total number of allele for that gene in a population. So remember the difference, number of copies of a particular, only particular is mentioned here. In a population, there are different types of alleles. So particular allele in a population upon total number. See here from this diagram, it is clear. Orange color moss, they are shown here. White color are shown. Then brown color. So different colored moss are shown. It shows about particular allele in a population. This is population. Population of moth. Then it is upon total number of all alleles. So these are all alleles, total number of all alleles for that gene in a population. So you have to remember allele frequency. Allele frequency is equal to number of particular allele in a population upon total number of alleles for that gene in a population. Genes are arranged linearly on the chromosomes. Yesterday also I had explained you about how these genes are arranged on chromosome. Suppose, for example, on this chromosome, this gene, for example, gene A, then gene B, gene C, gene D. So all these genes are arranged linearly. They are arranged linearly on chromosome. According to Mendel, every gene that influences trait has two alleles. We had already studied about this. That is, that influences trait. Trait means, can you tell me what is the meaning of trait? Girls, answer my question. What is trait? Trait is a character that has two alleles. Can you give me the example? Before few minutes only I have explained you, for example, height of the plant, it is a trait. Tall, capital T, small t, dwarf. So every gene that influences a trait has two alleles. So these are the two alleles of each trait. Which trait? Height of the plant tall, height of the plant dwarf. So these are the alleles. Gamete produced by individual furnish a pool of genes of next generation from which the genes will be selected. Any change in the gene pool affects population. So if there is change in the gene pool, population will be affected. You have to remember this. As you will study further, your concept will be more clear. So any change in the gene pool, this will affect the population. Gene pool changes due to migration of population and replacement of one generation by another. You are going to study in detail in the further slide. So gene pool changes due to, what are the reasons in the changes in the gene pool? Migration of population and replacement of one generation by another. Modern synthetic theory comprises of three main concepts like genetic variations, natural selection, and isolation. Now see about genetic variations. The change in gene and gene frequency is known as genetic variation. Underline this sentence from the textbook. Change in gene and gene frequency is known as genetic variation. See here in the diagram, it is shown. Mutation, we had studied mutation. Mutation is the sudden change which occurs in gene or in chromosome, due to which there is change in the phenotypic character. See here, this insect which are shown in green color. Here one insect which is green color, which shows green color, while other brown color. So why this mutation has been occurred, or why the changes has been occurred? Due to changes in the gene or due to change in the chromosome. Next is gene flow. So two populations are shown in this diagram. Few of them has been migrated towards another group of population. So this is called as gene flow. There are different reasons for the gene flow. We are going to study them. Genetic drift. See here in this diagram, they had shown this genetic drift. Some of the individuals from this population and they had been migrated towards this population due to certain accidents or due to certain changes, they have been destroyed. This is called as genetic drift. Natural selection, we are also going to study this natural selection with the help of example Biston betullaria in detail in the next slide. So remember these 4 points: mutation. Mutation means sudden changes which occurs due to which genetic variations takes place. Due to gene flow also genetic variation takes place. So see in this diagram, it is very easy to understand if you will carefully observe this diagram. Gene flow, it is shown clearly. Genetic drift is also shown. So if there are geographical barriers or different types of barriers, genetic drift takes place and natural selection is also there. There are 5 agents of evolutionary change: mutation, gene flow, non-random mating, genetic drift, and selection. 5 agents of evolutionary changes. Which are they? Or variation, genetic variations are also occur due to this mutation, gene flow, non-random mating, genetic drift, and selection. From this diagram or from this slide, it is very easy to understand. Here, we are guanine, thymine, guanine, cytosine, they are shown due to ultraviolet light. You can see here UV light. There are different agents, mutagenic agents. Some are physical agents, some are chemical agents. Physical agents like alpha rays, beta rays, gamma rays. So they, with the help of these rays, there is mutation in gene or in chromosome. Or due to chemical mutagenic agents, there are different chemicals. For example, mustard gas is a chemical which is used to change the gene sequence. So here, due to changes in the gene, mutation takes place. It is shown here. Then gene flow is also shown here in this diagram. That is, pollen grains are collected from with the help of this bee, and this honeybee, when it settles, or this insect goes to another flower, visits another flower to disperse the pollen grains on the stigma of another flower. Automatically, what happens? The genes which are present in this pollen grain, it is transferred to another flower. So gene flow also takes place here. Non-random mating, there is no frequent mating among the individuals of the same species. That's why it is called as non-random mating. Genetic drift, due to flooding, due to mountains, due to landslides, here genetic drift takes place. So certain barriers are there. Due to these barriers, population gets separate from each other. See in this diagram, it is shown very clearly. The barriers may be flood, the barriers may be formation of river, it may be formation of mountain, or due to landslide, the group of individual from the population get separated, and it leads to genetic drift. Then selection, certain organisms select the certain species only. This is called as selection. So we are going to study them in the next slide also in detail. So don't bother if you do not understand, or if your concept is not clear, it will be cleared in your next slide. So as you study further, your concepts will be more clear to you. Now see gene mutation. Sudden permanent heritable change is called mutation. Mutation can occur in the gene, in the chromosome, and in the chromosome number. So where it can occur in gene, in the chromosome, and in the chromosome number. Mutation that occurs within the single gene is called point mutation or gene mutation. And when the mutation occurs in the chromosome, it is called as chromosomal mutation. So what happens due to mutation? Phenotype of organism causing variation takes place. So we had studied this. See, gene flow. Gene flow is the movement of genes into or out of population. Gene movement may be in the form of migration of organism or gametes. For example, dispersal of pollens, which we had seen in our previous slide. Dispersal of pollen or segments of DNA, that is transposition, transformation. Gene flow also alters gene frequency, causing evolutionary changes. So due to gene flow, what happens? Gene frequency is altered, and due to alteration of gene frequency, what happens? Evolutionary changes takes place. Please note down this point or underline this sentence. What happens due to gene flow? Alteration in the gene frequency takes place, and due to change in the gene frequency, evolutionary changes occurs. Genetic recombination. In sexually reproducing organisms during gamete formation, exchange of genetic material occurs between non-sister chromatids of homologous chromosomes. So, with the help of diagram, I will tell you what are non-sister chromatids and what are sister chromatids. So during meiosis, you had already studied mitosis and meiosis. These two are sister chromatids. One chromosome is paternal, another one is maternal. These are called as homologous chromosomes. So when there is exchange in the segment of DNA, suppose this non-sister chromatid overlaps with this non-sister chromatid. These two are non-sister chromatids. This is called as crossing over. It is called as crossing over. They overlaps upon each other. It is seen in Pagetin stage of meiosis 1 of prophase 1. Prophase 1 has different stages like liptotin, diplotin, zygotin, Pagetin, dikinesis, different stages are there. So during this stage, crossing over occurs. So due to crossing over, recombination of genes or exchange of genes takes place from this patch of chromosome. The genes are transferred to this chromatid, and in this way exchange of genes takes place. So do you got the difference between sister chromatids and non-sister chromatids? These two are non-sister chromatids. Yes, these are the non-sister chromatids. So crossing over takes place between non-sister chromatids of homologous chromosome. These two are homologous chromosome. So you have to remember this point so that it will be easy to understand. This is called as crossing over. It produces new genetic combinations and it leads to variation. Remember this, only in sexually reproducing organism crossing over takes place, and Due to crossing over, new genetic recombination takes place, and due to new genetic recombination, variations occur. That's why each and every individual is different from each other, except homozygotic twins. You know, homozygotic twins, both the individuals are male or both individuals are female. Why? Because they are obtained from the single zygote. Heterozygotic twins, they may be either uh, female or male child, or they may be, both may be male or both may be female child. So this is heterozygotic. Why? Because different zygotes are there. Except homozygotic twins, all other individuals, they are similar to each other. So as the genetic recombinations takes place, it leads to variation, and variations, it is very essential, or it is variation, it is due to sexual reproduction. We cannot see variation in asexual reproducing organisms like fission or fragmentation. There are different types of asexual reproduction. We cannot see this type of recombination. So you had studied in your previous topic difference between asexual reproduction and sexual reproduction. Fertilization between opposite mating gametes leads to various recombination. So there are different recombinations resulting into phenotypic variation. What is phenotypic and genotypic? Can you tell me? What is phenotypic and genotypic? Girls, answer my question.
Phenotype is the external appearance of an individual for any trait.
Okay, very good. What is genotype?
Genotype,
Huh?
it is a makeup of an organism with respect to a particular trait.
On the basis of genetic constitution. Yes, while studying Mendel's laws, we had studied phenotypic and genotypic ratio. Do you remember?
Yes,
While
ma'am.
writing phenotypic, we used to mention their tall. And for genotypes, we used to write their alleles, capital T, capital T. Yes, this is the genotype. When it is based on the genetic constitution, we call it as genotype. When we tell them on the basis of external appearance, we can see this plant as tall. So this is the phenotypic character. We can see this plant as dwarf or short. We can tell this on the basis of phenotypic appearance. So this is the difference between phenotype and genotype. Resulting into phenotypic variation causing changes in the frequencies of alleles. So this is about genetic recombination. See here, it is clearly shown and it is very clear, easy to understand when you will watch this slide. These are non-sister chromatids. Crossing over takes place in these non-sister chromatids, and here exchange of genes takes place. Due to exchange of genes, what happens? Recombination takes place. Due to recombination, variations takes place, and variation leads to evolution. And it occurs only in Which type of reproduction? Girls, variation occurs in only which type of reproduction?
Sexual reproduction.
Yes, but very good. Only in sexual reproduction. Why? Why? Tell me the process. Can you tell me the process? Why variation or evolution occurs only in sexually reproducing organism? You know, in sexually reproducing organism, gametes are formed. And gametes are formed only by meiosis. Yes? During meiosis, crossing over takes place. See here in this diagram, crossing over is shown. Exchange of genes occurs. Due to exchange of genes, recombination takes place, and due to exchange of genes, variations occurs, and due to variation, evolution takes place. This is tetrad. Tetrad is shown here. These are homologous chromosome. One chromosome, it is maternal, another one is paternal, and it takes place, crossing over takes place between non-sister chromatids. These are two non-sister chromatids. Genetic drift. Any random fluctuation or alteration in allele frequency occurring in natural population by pure chance, underline this sentence. By pure chance, it is called as genetic drift. For example, severely reduced, the size of the population is severely reduced due to natural disasters. Which are the natural disasters? Like earthquakes, floods, fire, then landslides. So these are the natural disasters. It causes elimination of particular allele from a population. We have already studied this in a previous slide. So due to natural disaster, which are the natural disasters? Earthquakes, it may be flood, it may be fire, or it may be landslide, eruption of volcanoes. So there are many different natural disasters. And due to this natural disaster, elimination means removal of a particular allele or particular individual from a population takes place. It is called as genetic drift. Genetic drift is also important factor for evolution. See here in this slide, it is clearly shown. Change in allele frequency due to chance event. So these are the chance event. The ratio is 5:5. Then it is changed to 5:1. See here, these blue color beetles, they are removed due to this natural disasters, and the ratio has been shown to 10:2. Because reproduction has been occurred only in this red moth. So the ratio has changed. It is called as genetic drift. Due to genetic drift, allele frequency also changes. Smaller population have greater chance for genetic drift. It will result in the change in the gene frequency. Genetic drift is an important factor for evolutionary changes. Here also in this diagram, in this slide, chance event has been shown. This green moss, they had been destroyed. These green beetles, they have been destroyed due to different barriers due to different natural disasters, and here in this population more number of brown colored beetles are shown. Green colored has been eliminated from this population. So due to this, evolutionary changes occurs. Chromosomal aberrations, the structural morphological change in chromosome due to the structural and morphological change. Morphological means on its external structure. Yes, morphological change in chromosome due to rearrangement is called chromosomal aberrations. Different genes has been rearranged on these chromosomes. It is called as chromosomal aberrations. It changes the genes arrangement, order or sequence that result in variation. So due to chromosomal aberrations, variations occurs. Which are these chromosomal aberrations? It is very easy. See here, deletion means loss of genes from chromosome. Duplication, genes are repeated or double in number. Inversion, a particular segment of chromosome is broken and gets reattached to the same chromosome in an inverted position. So in inversion, what happens? Same sequence has been reattached, but this position is inverted due to 180-degree twisting. There is no loss or gain of gene complement. Translocation, that is transfer of part of chromosome or set of genes to non-homologous chromosome, is called translocation. We are going to study with the help of this slide. See here, it is very easy to get your concept clear if you will see this slide. A, B, C, these are the genes. D, E, F. So this is the centromere. These are the arms of the chromosome. While studying deletion, D is deleted here. See here, only A and C are shown. So this is deletion. Duplication, here gene B is duplicated. A, B, B, C. So B is duplicated. So this is duplication. Due to 130-degree, 180-degree twisting, here inversion occurs. A, E, D, C, B, F. So this is inversion. And translocation, A, B, C from another chromosome from non-sister chromatid J, K, L has been attached. While from this D, E, F is attached to another part of the chromatid. So this is called as translocation or transposition. So position of genes has been changed here. From non-sister chromatid JKL has been shifted to another non-sister chromatid, and DEF, they had been shifted to another part. So you have to remember deletion, duplication, inversion, and translocation in chromosomal aberrations. Natural selection. According to Darwin, natural selection is main driving force behind the evolution. Genetic variation drives within the population. The fittest will be at the selective advantage, more likely to produce offsprings, than the rest will enjoy greater survival and reproductivity.